guys. We are going to learn about translation today. Thus far, if you're looking at kind of big picture of what we're doing right now, I showed you this yesterday. You're going from DNA to RNA, RNA to a protein. So we covered DNA already and DNA replication. So DNA just making more of itself during that S phase of interphase when cells are undergoing cellular reproduction. Then going from DNA to RNA, remember that's that transcription piece. So just kind of opening up that strand of DNA, taking a small segment of that and making the mRNA copy from it. That's transcription. So now we have that single-stranded mRNA. Remember, G's and C's always line up. A's line up with a U now instead of a T. There are no T's in RNA, only T's in DNA. Today we're going to talk about translation. So how do we go from that single-stranded mRNA to a protein? How is that done? So just keep this big picture in mind as you're going through this whole process. All right, so translation. We are now, so just think of that mRNA strand, that single-stranded mRNA that was made from the DNA inside the nucleus. It then heads out, once it's made and disconnects, it heads out into the cytoplasm to get read, or we call it translated. So hence the step of translation. That RNA strand has to get read or translated. It's read 3 bases at a time, so 3 letters at a time. And we use either this codon wheel, it's a really colorful wheel over here, or this codon chart. You can use whichever one is most comfortable for you. I think it's easier to use the wheel personally, but if you want to use the chart, that's totally fine. That mRNA strand is read 3 bases at a time, and then those amino acids that are made are going to string together to make a protein. So let me just give you an example on this next slide. Okay, so let's just look at the visual because I think that's easiest. So if this strand is your mRNA strand, your single-stranded mRNA, every 3 letters, every 3 bases here makes a codon. So in this case, GCU is codon 1, just the first codon. A codon is simply a 3-letter sequence. ACG is another codon, it's the second codon. GAG is the third codon, continues on. Each of these 3 base codes, or each codon, codes for an amino acid. So this first one is GCU. So if we go back, and I'm going to use the wheel, you start from the middle of the wheel and you work on out. So G is here in the middle, next out is C, next is U. So GCU codes for this amino acid on the outside, ALA, which stands for alanine. So codon 1 is alanine. And then you just keep going. The next set of 3, the next codon is ACG, which you go to your wheel here, start in the middle, working your way out. A is up here. What was it? ACG, okay. A in the middle, C right here, G here, so that is THR, just a 3-letter abbreviation for threonine. Third one, GAG, GAG, GLU is the abbreviation for glutamate. So each 3-letter sequence is called a codon. Each codon codes for a specific amino acid. You'll note that, sorry, I keep going back and forth, but you'll note that there are a lot of amino acids that have more than one codon that codes for that specific amino acid. So for example, threonine, ACU, ACC, ACA, ACG, all 4 of these different codons all code for the same exact amino acid. If you're using the chart over here, first letter is on this side, the left side. second letter is on the top, and third letter is on the right-hand side. So if you're going CCA, for example, you'd go C over here to C up here to A alongside over on this side, and where those three meet, that's where you'd have your codon, and that would be proline. You'll also notice that there's one start codon, which is AUG. There's only one start. It's the MET is the abbreviation for it. The stop codons, there's three of them: UAA, UAG, and UGA. So just make note of the start and the stops as well. So that's really how that mRNA strand is read or translated into amino acids. The string of amino acids, if you remember back to macromolecules, that string of amino acids then winds up, folds up, and it creates a specific protein. So this is really just the blueprint for a specific protein that's going to be made. Okay. What that looks like in your cells, this blue or teal structure is the ribosome, so that is the rRNA. The mRNA strand, the messenger RNA, is the single strand here that's going to be read into 3 bases at a time to make your amino acids, to make your protein. And the tRNAs are these, again, key or cross-like structures that carry the anticodon and the specific amino acid that codes for that codon. So for example, you have ACG that's being read on the mRNA. The anticodon is just the opposite letters. That's what the tRNA is holding, the anticodon. And it carries that anticodon over, it matches up and lines up with the mRNA bases, and it carries a specific amino acid that's made for that UGC. Those amino acids then link up together up here to make your protein. The job of the ribosome is really just to hold the mRNA in place as it's being read. Okay, so you've got your rRNA, I'm sorry, that's holding the mRNA strand that's being read, the tRNA that's carrying the anticodon and the amino acid. Okay, so here's your sequence. You have your transcription, you have your translation, and then protein synthesis. This diagram on the left is showing all 3 of those steps and the specific location in which it's happening. So you have inside the nucleus, you have your replication happening, just A's and T's, G's and C's lining up. You have the DNA being transcribed into the mRNA strand. Once that strand is made, and you'll note the U's now, that mRNA strand heads out into the cytoplasm to be then translated, read, by the tRNA in sets of 3 or codons into these amino acids. You got 3 of them down here to make a specific protein. Just make note of, maybe I can move that, yes I can. The difference between the codon, which is on the mRNA, that's the one being read, and the anticodon with the opposite bases, pairing up or lining up to create that specific amino acid or carry that specific amino acid. Okay, let me stop the video here and then we'll do some practice in a second. Okay, so I'm back to just do a little practice with you. I'm going to give you a strand of DNA, and then we're going to work through the mRNA, tRNA, and then amino acids. So we're just going to run through the whole process here. So let me try and hold this up. Should not give you opposite. There we go. Oh, this is tricky. Wow, I feel like I'm really awesome at this. All right, so you see it maybe? So DNA is, we have, what do we have? TAC, TCG, CGG, if that is our strand of DNA. we first need to make the mRNA strand from that, the messenger RNA. So if we go from DNA to mRNA, remember we're matching G's and C's. A's go to U's, T's go to A's. So let me just rise up. Here we go. So you'll note we made our mRNA strand now. T matches with A goes to U, C goes to G, continues on. That mRNA strand heads out into the cytoplasm so that process right there, DNA to mRNA, remember that's called transcription. Then we need to translate this code, this message into our amino acids to make our protein. So we're going to go from mRNA to tRNA, which is that tRNA is the molecule that's coming along and reading the mRNA 3 bases at a time, or each codon, and it's carrying just the opposite letters and the amino acid that codes for that specific codon. So there's our tRNA, and then from there, you always look back to your mRNA strand to make your amino acids from, or to get your amino acids. So looking at your mRNA, AUG is the first 3. So you find that on the codon wheel or chart, whatever your preference is, and AUG is met. Can you see me? Maybe not. Here we are. ACG codes for the amino acid serine, and GCC codes for alanine. So note that the mRNA is the strand that's being read for your amino acids, and the tRNA is really just the code that unlocks that specific amino acid. So it's just used for unlocking the amino acid. So there's one example for you. Okay. Now, one last thing. In your assignment here, let me find it, which has been posted, you guys started doing, I don't want to show you this too close because it has all the answers on it, but you guys, this one here, you guys did I had you do the DNA to the mRNA strand and the tRNA. You can now use that mRNA strand to write down and find your amino acids. So use the codon chart or the wheel from the notes here to complete the transcription translation assignment. All right, let me know if you have any questions on that. Either email or chat with me and we'll get that all straightened out. All right, hope you're having a good day.